star turns up really abundantly in collections. You know, there are sometimes when I get 6 to 12 of these in a bucket, and we've, I think, only seldom seen them alive. You know, sometimes when I've seen them in the Atlantic, they're orange, and this one is white. So that is interesting. They form kind of a species complex across the Indo-Pacific and with one species in the Atlantic. So there's an interesting biogeographic question there as well. We don't know anything about their biology. So seeing it just on the hard bottoms like this, and I think this is their lower end depth range, is quite interesting. So there's a whole bunch of mysteries that just looking at one, you know, sort of emerged. But for the moment, I just thought I'd let you know that it was noteworthy simply because, you know, we see a lot of them in collections dead, and very seldom do we actually get one alive.
Great.
So thank you for stopping for it.
Thanks, Chris. The coral folks on the line, can you tell us what we are seeing on these sponge spicules?
So Shirley, these projections coming off of the sponge are actually spicules. They're part of this particular species.
Right, but there's something. Looks like there's something growing on them.
Specific name, and I've forgotten that. I'll have to go look it up.
Boy, but it looks like they're polyps.
That's all sponge except for that brittle star that's on top.
Okay, so the things that I'm thinking of that are looking like polyps on those spicules are actually part of the sponge.
Video's clear, so we can look
and I'm going to go try to find the names of what those parts are called. It's in Systema Periphera, and I've got that on my iPad, so I'll take a look.
All right. Okay.
Clear.
All right, all set with this watch lead?
All set, thank you.
All right.
Okay, I give you your camera back. And the laser's back on. Want to get another move going then? Yeah, you're welcome.
Yeah, what direction you guys want to? You guys?
I guess still going the same way, right?
Yeah,
they want to keep going until we run into sediment only.
That's a good direction.
Yes.
We could do.
Yeah, it all seems fairly flat, so we can just keep going the way we're going, I guess.
Okay. 310. Bridge, RV nav.
RV nav, this bridge.
Ready for a ship move. 30 meters, bearing 310 degrees, decimal 2 knots.
I copy 30 meters, 310, 0.2 knots.
Good copy.
Yeah, my sonar is just a collection of hundreds of rocks.
Boulder City.
I still think we have a little compass offset. I still keep ending up. See, I'm in the right side of your screen again. So I guess when we call it a move of 310, maybe you have to face like 320. And then you'll stay aimed at me.
Okay, we can try that, yeah.
Does that make sense?
It's a big boulder over there to the right, some big one, big rocks.
Well, for that move, you sort of went off to starboard and then came around a little bit.
We
so
have
maybe.
Oh, you don't think
series of real hard returns, both to the right and to the left. Because it seemed to be tracking
before pretty well when we were moving, you know.
Yeah.
And of course, the rock right in front of us is making a nice sonar shadow. Isn't that cool?
Oh yeah.
I guess it could just be the current too.
So all it says, at least in the diagnosis for Walteria, and that's that sponge we were just looking at, is distal lateral processes are simple or dichotomously branching outgrowths of the lateral wall.
Yeah, I'm looking at my systema. I'm looking at my systema peripheral right now too, Chris.
But
it's very weird.
They did seem to be rough surfaced, the spicules.
Oh, Don, I don't know if they
Reading
passed
over
it on,
Shirley's
but
shoulder here.
we're going to keep the port slider inboard a bit.
She's got an
since
article
the
with
shilling
pictures
has
of
sagged down.
sponge spicules.
Oh, okay. No,
This
they
is the
did not.
sponge
So
Bible.
it should
Are
be
you
in a
kidding
few
me?
inches, I think.
Okay.
Yeah, it is.
Yeah. Oh, actually.
Oh, I see. Yeah, the shilling did.
Yeah.
Shoulder that came down. Huh.
And I guess it's...
So they call them crossdalia too,
Why?
I think, and
They
they
hadn't
say
used it, right?
that
They
they
just
can
turned
does the lock
protrude
valves
outside
and
up
they go
to
down the
14
imbalance.
millimeters long.
Okay.
Same thing that...
But nobody ran it.
No.
No, okay.
Yeah, just the imbalancing pressures.
Okay.
Video, can we get a quick zoom?
Okay, so here's some chrysogorgia here that I think we're going to want to take a closer look at as well. So
that
the
one's dead.
bushy corals
That's
over on the
unusual. Okay.
lower right
Okay.
and the one over on the left, please.
And that, if I'm not mistaken, the one that we just zoomed on, is that a black coral? The
Not
stems looked black, but it didn't look alive.
Okay, video, what's going on this coral?
Roger. These are so
oh
pretty.
my gosh.
They really are nice looking.
Little eyes poking out.
So now that I see this, it reminds me of the new genus that we were looking at the other day when you
I have to
explained
take off
that
here.
there were
Again.
upward curving branches. So now I can see how that one that we saw the other day is very different from this one where the branches are not upward curving. Very interesting. We're going to try and get in again.
Okay, you can start going in. Co-pilot, will you take my tilt?
Certainly. Okay. Coming up.
Yeah. Okay, video.
Okay. Tell me when if you want to go up some more.
Maybe just a touch up. Just
Touch up.
Oh, yeah. Let's see a squat lobster in there. And
What is
an
the
anemone.
more
It looks like it might be an
globular-
looks like it
Yeah,
might be
looks
an
like it's-
anemone. I'm not sure. Yeah, Diva says an anemone. Nice, very thick branches. Les
Okay,
says it
whenever you're
looks
done
like
video.
one of the ones they've been studying from the Atlantic. Okay, and when we're done with that, I think there was a black coral, and then there was another one of these well, I don't know if it's the
The
same
ship
kind
just
of
got in
chrysogorgia.
position, so I got about
Okay.
5 or 6 minutes left to swing.
Okay.
Okay. Move on soon.
There
So
was a
this
black
one-
right in the. There was a black
Okay.
coral, I think, right
Sticking out.
over to the right.
Give your
Yeah,
camera
just
back.
above
Sure if you
the previous coral.
The dead one or the one beyond it?
I'm wondering if Tina wants to look at the dead one.
I think she wants to look at the live one that's
I
in the
would
back.
hope.
Yeah.
Okay.
Attention on the Okeanos at this time. The friendly group of engineers is going to be pouring bugs down the drains of persistent showers. Thank you for your cooperation.
Okay. Free to zoom video.
Ah, yeah. Ooh, and what is on the stalk in front?
I'll just hold here so we can get both.
Let me know if you start passing over me, co-pilot.
I will.
we need more zoom for the ID, or is that...
No, we're
yeah.
good. We're good. We're
Okay.
good. Thank you.
Roger.
All right.
So
And-
anthids, says Scott,
Yep,
on the
yep.
stalk in front.
And Carl, I don't know if you're able to, if you have time, if you can look at one of those other kind of bushy, Chrysogorgid
Yeah, we-
corals.
Copy.
Okay, thank you.
I
Take a
will
quick
talk
look.
to
Just
them
in front.
about,
Yep,
the
we can
fact
do
that all
that.
this goes to public.
And the black manganese encrusted material
All right.
projecting from the
Zoom in
boulder
there.
is,
Actual snap zoom.
Maybe
Actual
a dead
stuff.
sponge. Maybe.
but really big one. You said,
Yeah.
I forgot
Big
what
one.
you
We're
told
looking
us
down
it was
80%
somewhere
well,
in the
I
dive.
don't know. I mean, it's just a
To
wild
move soon?
guess.
Okay.
These barrel sponges, but I don't
All
know.
right. Let's go wide video.
Okay.
I mean, those barrel
Yeah,
sponges
the
do
current's
occur in
picking
the
up quite a bit.
Pacific for sure.
I took off.
Yeah, you can see the
just-
But not
particles in
deep
the water.
water.
moving.
Oh, okay.
So it would have had to have been a very long time ago.
All right.
Well, it is pretty heavily covered with manganese.
310 was the lead.
310 was the choice, yeah.
All right.
And ship has stopped moving, so
Ship's been stopped for about
5 minutes.
stopped at 53.
53.
So 3 minutes.
3 minutes.
And there we go.
So how much longer do we have in this dive, Shirley?
We have about, let's see, 1 o'clock, 2.5 hours,
That's good.
right?
Yeah, we're about
Yeah,
30
about 2.5
meters
hours.
below the top of the wall that we're climbing up. A little less
I
than
think
that.
we should initiate another move, huh?
Sounds good to me.
30 meters.
As good as any, yeah.
310.2. Bridge, RV nav.
RV nav this bridge.
Ready for another ship move, same as last. 30 meters, 310 degrees, decimal 2 knots.
Copy. 30 degrees, 3100, decimal 2.
That might be a fish on the
He
right.
said 30 degrees. I
Yep,
think he meant
there
30
is a
meters.
fish.
This is just,
Yeah.
you see a fish?
Bridge, go ahead and copy the range on that move.
There he is, on the
Yeah,
right
30
middle of
meters.
the screen.
30 meters high.
You look at the fish if we can,
Good copy. Thank you, Bridge.
up to it.
Okay, video. You can
Well,
zoom.
it's moving over toward that. Is it a sponge? To the left?
Yes, it is. Hexactinelle sponge. This is a nice view of this fish.
I'm going to take a touch more and hold there.
He's not very well behaved. Kind of
Prefer to
swimming
show us his tail.
up and away. Let's try one more zoom on him, then we'll go back.
Roger. Nice.
Calling this Aldravandia. This is calling it Old Blue Nose.
Yeah.
You can see that.
The head looks almost translucent. You can see its little tiny brain up there, maybe.
Okay. Let's
Just let him
swim
fly
away.
up. And I'll hold this. Nice. Coming full wide.
All right, back down.
Nice. Good job.
Was there any interest in that sponge that we passed over?
No, that's okay. We know what
Okay.
that one is. Thank you. We've recorded it for our notes. Thanks.
Yeah, Tina says this is her favorite fish.
Is it our favorite fish for
Lots of that
today?
rock
Saying
there.
Or
I'm not
favorite
sure.
fish?
Boyd.
Ever.
Ever.
We do
Yeah.
not
Oh, I
know.
see. Yeah. Was she there?
No.
She is
I'll
saying
come
it
down
looks a
here
little
and then
bit
I'll
like
move
a
ahead once
blue
you
fox.
follow me.
I can give you some more leash here. Go ahead.
All right, I'll go to starboard and then do a survey to port to see a bunch of stuff.
So you just push to starboard and let the current bring you back to port. Just kind of.
Well, the good news is eventually we get to turn to the north.
I think we can skip the lyrate coral on the right, but the bushy coral that's sort of center right now, I'm not sure we've seen that so far this dive.
Okay. Did you catch that, Carl?
The one on the left side of the boulder. He's turning the wrong way, the other left.
He's getting there now.
This could just be another Paragorgia, so it'd be another false alarm, but just making sure. Like now, the closer we get, I think it probably is. But
okay.
We're here.
Yep. Looks like another Paragorgian.
Still interested in zooming?
Shirley likes bubblegum.
I do.
Yeah, we could probably pass on this. We got all types of images of these guys.
Okay.
Did you catch that, pilot?
Shore votes for a pass.
We'll do, as long as I'm here, we'll do a quick 10-second for video.
Roger.
Do you want to go in?
All right. Just hold here.
Yeah.
Well, maybe we'll go just a touch more.
You want lasers off?
Yes.
Yes, please.
Thank you. All right.
Okay.
Thank you.
Okay. Lasers back on.
Now we'll do a little drift. Co-pilot, can you check that the drawer is all the way in?
Roger that.
I think when I bumped up against that rock, or when I almost bumped up against the rock, I saw it's out a little.
So we still have about 30 meters to go to get up there. It looks like we'll have plenty of time.
Came back in a little bit.
We're doing well on our
good call.
dive track.
And there's a floater. And there's a something on that something on that rock that looks like a sponge, Shirley.
I don't know.
Is it a lobster?
Looks like a lobster.
Take a closer
Rock
look.
lobster.
Oh, man.
Take a closer look.
This is some sort of a fish or a lobster.
It's a rock lobster.
And
Good grief.
Something squishy there.
Oh, yeah.
Oh, and there's also something projecting out of the rock on the lower left below the pink thing.
Okay, video. Go in.
What are we looking at? Oh, it's one of those lobster things. Wow. These are really pretty. They're also pink. Not a big fan of pink, but I like the critters.
Quite intricate.
Okay, folks.
Polychaeid. Polychaeid or polychelid?
We tell two
We have two different spellings
interesting.
here.
It
We have a
puts his legs away.
polychelid that
Just kind of hangs
Or
there.
no,
I guess it makes it streamlined.
Polychaeidae.
Yeah, he's probably holding on with them
Maybe
too.
it's a polychaeidae, polychaeid.
lobster. For those
Yeah,
of
it's
you
a
who know,
lobster.
lobster.
I guess main lobsters go down into cracks to get out of the current.
It has very fuzzy ears.
And have pretty good defensive weapons too.
Okay, then we'll just go down to this guy.
Have we seen any of these guys with their claws out? Do we know what they have?
Tunicate.
I've seen a few, and I think they were all like this.
All like that.
What's this guy?
Yeah. Okay, video. It's pretty cool.
Wow.
What's he saying?
Happy 4th of July.
Probably.
Tilts up a touch there.
Oh, it's got a little polychaete worm on it.
This is really a bizarre creature.
Very bizarre tunicate. Yep.
Yeah, we
Transparent.
saw one earlier.
Yeah. I think this is
Just
my favorite,
exactly
one of
the
Deb's
same
favorite
thing.
animals. Polynoid polychaete on it.
Ship has been in position for about 3 minutes. You got about 2 or 3 minutes left to swing.
Okay.
And start thinking about the next one.
You want to check with watch lead if they still want to continue?
Yeah, we still want to continue.
Yeah.
Along this track?
Yes,
Yeah.
we do.
Okay, yeah, we'll continue along this track.
It's another big patch of sediment with the rubble in it.
And it has.
Has ripples.
Thank you.
It has
Very good.
ripples, and it has dead sponge stalks coated with manganese and partially covered by sediment or broken into small bits. I would vote for the former.
Boy, I'll say, look at them all. Everywhere. At least
There's a mass
right
extinction
in the bottom.
event of
Well,
these.
yes, like just in the bottom half of the screen, and then above that there are fewer.
We were talking about this earlier in the chat, and Les proposed a large settlement event of these sponges, followed by growth and then senescence with no new recruitment. Must have been amazing when they were all alive.
Yeah,
This
real
big,
forest.
I mean, really big.
Very
Oh,
mature.
you see something.
Whip-like on the rock.
Indeed.
Okay. We're both freezing.
Nav, I'm sorry, did you call in a move, or are we still waiting?
No, we're still waiting. You've been stopped for 4 minutes now. Almost 5.
Probably should get it rolling.
Yeah, good call on it.
Okay.
Don't see much going on
So
this
this
stuff.
looks like it's dead.
Pretty job, Nav.
Or been chewed on,
Yeah.
thoroughly chewed on
Yeah.
by something else.
Now this bridge.
We're ready for another ship move. Same as
Okay,
last.
yeah.
30 meters, bearing 310 degrees, decimal 2 knots.
Okay, I copy 30 meters.
So maybe this is
Oh,
another one
man.
of the
decimal 2.
It's a good
Aplicopha
copy,
just chewed its way all the way up.
Sure isn't much left on this guy.
Tara Luke says there's something living up near the top.
Yeah, I saw that too. We
So
will get
long
there.
Segments.
Mm-hmm.
There it is.
Mm-hmm. Looks like a teeny tiny
Crinoid.
crinoid.
Did he eat this entire coral?
No, I don't think so.
Okay, want to get a zoom on him?
Sure. Are those gastropods below it? Or some
A touch more.
Not tell.
not sure.
Tilt down.
Do you think those are gastropods, Shirley?
I don't. It sure does look like one.
I don't know. Yeah, I'm looking down here.
Yep. It's another aplecophoran. How can something that little have eaten all
that much?
That much. It must have taken a while.
And Scott's going, Shirley, you suggested an aplecophoran.
Yeah, right.
Polished this off. Interesting idea. If so, I wonder how long it took.
Yeah, probably
years. Well, how come it's not bigger?
Or is it
dead now?
No, it doesn't look like didn't look like it was dead.
Well, it's very much smaller than the one that we collected.
That's right, and it's
white.
Yeah.
So maybe it's just segments of it after it
fallen
starved
off.
itself to death. After feasting on the coral.
Yeah, so then what happens? Does it like crawl back down and find another one, or just...
you're asking me.
All right, I'm going to have to go
That's a forlorn
I'm going to have to go look up something about
the
life history of aplecopherans. Les Watling was saying that they looked at sea stars on a Paragorgia that fell over in an interval of 18 months, and the sea stars had hardly moved at all.
Really?
Mm-hmm.
Wow. Well, I guess you're
so this is something that Chris Ma observed as well. The aplecophoran probably reproduces and dies.
Yeah, there were some big sponges here.
Mm-hmm. Indeed. That would have been pretty interesting.
It sure would. Maybe we should indeed have dived on the southern side of the fault.
Well, let me ask you a question.
Okay.
How many millions of years ago would this have been shallow?
Good question.
80, 90. I mean, I'm just trying to think.
Yeah,
I'm trying to think what would have been on a shallow reef, because we were seeing these things that we were suggesting, wildly guessing were barrel sponges, which are in shallow water. I mean, these are shallow. They don't. I mean, they may go down to 50 meters, but not any deeper than that I've ever seen at least in the Caribbean. So I'm trying to think what kind of stalked sponges would have been in shallow water way back then too. I don't know. We are coming to you live from NOAA ship Okeanos Explorer, and we are looking at a seamount called Friar Guillot and the Mariana. Guillot. Is it a silent T?
It is indeed a silent T.
Guillot, without the T.
That's right.
I thought there'd be someone living on top of this rock.
We're in the Marianas Trench National Marine Monument.
We are. Western Pacific, where seamounts come to die.
They grow. They are born. They gradually sink.
And then they get
abducted. But before they do, we get to look at their fossils.
We sure do. Yesterday's dive was on an exposure of the reef on the western side of
Carl, can we
this
look left,
particular seamount.
Look left.
Yeah.
And we just climbed up and up and up, sequence after sequence of reef and really fine carbonate layers.
There's two bushy corals over on the far left. I don't know if you can move over there or not.
I could take a quick look.
Okay.
So on this side of the seamount, everything is covered with manganese oxide crust or coating, and that's why the rocks look so smooth, sort of rounded everywhere. That's not the real rock you're seeing, but the coating.
that big
Okay,
tall
video.
one.
Let's get a zoom on these. I am floating here in the current, so
Sounds good.
I'll have to excuse any movement. Coming up to them.
Okay.
Wow, these
So
are
give it
beautiful.
more. They look a bit like fireworks, don't they?
4th of July.
Very festive.
I think I'm getting pushed back.
Current's picking up, isn't it?
Yeah.
That's full zoom.
It's not too bad though. It's nothing like the shallow dives.
What do you have to put on you? So you're like at 22 just to remain stationary?
Yeah,
Okay.
forward. See, I came
Forward,
around to
so
this side,
quarter.
so I just face into it, and then the tether drags straight behind.
Yeah.
That way my heading doesn't wander. You know, if I tried to...
Watch it, go ahead.
If I tried to do the
Fight
same
it
shot
laterally,
from
yeah.
over here,
No,
it's
yeah.
not just the lateral, but the tether also turns your
Dive
heading.
and waypoint. The ROV is 260 meters away.
260. What's the time remaining on the dive?
2 hours, 10 minutes.
Oh, okay.
But one good thing for us is we're heading fairly into it, and we're eventually going to turn, so we should be able to kind of face it most of the time. All right, I'll drive
Okay.
back to center.
All right.
Les, you remarked that that close-up image that we got was really good, and it looked like the polyps were reproductive. Can you tell us how you figured that out, aside from the fact that you're an expert? I'm waiting for him to type it in. He's not on the phone line.
Okay. Oh, fish, two fish.
One fish,
Wow.
two fish.
One fish, two
Wanna
fish.
go in center
Pink
video?
fish and other
They seem
fish.
to be on a journey with the current, so I don't think we'll have a lot of time.
Yeah.
Come back up.
So the current is pretty strong from our right to our left. You can see white specks of material floating in the water column and moving from right to left.
Okay, you think seriously stopped?
Ship stopped 4.5 minutes ago.
Yeah.
Yeah, I think we're ready for another.
Okay. We'll probably make it to the waypoint a little early. It's all right.
Or
Yeah,
maybe
that's
we'll-
okay. If we
Oh,
make
you'll
it to the
find-
waypoint a little early,
Or
that's
we'll
fine.
stop
We can
and
spend
find some-
time looking around,
Yeah.
maybe going a little bit further.
Okay. Bridge RV nav.
All right, now this bridge.
Another move, same as last. 30 meters, bearing 310 degrees, 0.2 knots.
I copy 30 meters, 310 degrees, 0.2.
Good copy.
I'm getting pretty far out there, but I won't go any further than this.
Looks like another one of these Walteria sponges.
Might be a carnivore sponge or two on the left there
It
too.
might be.
Okay, want to go in on this one?
I'd rather go on the one on the left.
All right,
We've
we'll
seen
get
the
that
one on the
next.
right.
This guy?
Yeah. Oh, that guy. So yeah, hexactinellid sponge.
Yeah, we did see one of those earlier.
Actually, I thought
Lasers
Tara
off.
was talking about another one.
You can leave them on.
Okay. Leave them on for now. Okay.
Yeah, it's not the white background. I don't think.
It's not a beauty shot.
Yeah.
Okay.
More for ID.
The one over the far left.
Okay, so skip the one in the middle.
Yeah, we've seen that one.
The little round one or the stock?
The little round one's looks like an anemone. We can take a quick snap zoom of that.
Go in this one. I think it's a sponge.
Oh, no, it's not an anemone. It is, you're right, it is a sponge. See, that's another weird one. Another phoenix sponge.
Sponge urchin.
Yeah, sponge urchin.
Yeah, I don't think I'd like to touch it.
And now with those long spicules, that would be kind of painful, even through double gloves. That's kind of weird. It's like the other one that we saw that was more of like a different species. It was a vase-shaped sponge, and it looked like the sponge was growing up from the inside of an older dead sponge. Okay. Very weird. And there was something stalked.
Yeah.
That might be a coral.
Yep.
Do we want any closer view?
Yeah, do you have any more?
Yes, that's full.
Okay. Yeah, that one's pretty far.
Yeah.
Okay, whenever you're ready, we are.
All right, clear.
So we are on the NOAA ship Okeanos Explorer. This mission is part of a 3-year campaign. And Chris Kelly is actually kind of the scientist in charge of that campaign. It's called the Campaign to Address the Pacific Monument Science, Technology, and Ocean Needs, or Capstone.
Thank you.
Capstone was designed to address the fundamental need of our resource managers to better understand the deepest parts of the nation's marine monuments. In this particular case, we were really focused on this site because of the potential for deep-sea mining activities. Oh, okay, so we're seeing some big rocks here.
Reach over with
and
the craft
possibly
and push it back
precious
up.
corals, although it's a little bit deep for the precious corals.
Like
but we have seen some that are in the same
I don't
family.
think I can reach that far.
No, not quite.
But this is an area that's been identified as a potential site for deep-sea mining activities, so we wanted to be able to document what living resources there are here as well.
Oops. I just rose about 5 or 10 meters.
Partner, NOAA's Office of Ocean Exploration and Research has
take a look around
designed this mission. Plan is conducting the mission. The ship is operated by NOAA's Office of Marine and Aviation Operations, and the other NOAA partners in this Capstone campaign are NOAA Fisheries, NOAA's Deep Sea Coral Research and Technology Program, and the Marine National Monuments Program. Another important partner is the NOAA's National Center for Environmental Information, which makes it possible for NOAA to be able to share the data and images within 2 to 3 months after this expedition is over. And of course, the partner responsible for operating and maintaining and upgrading our ROVs is the Global Foundation for Ocean Exploration. And I'll have the guys, I think the guys introduced themselves a little while ago, but...
They did, but they can do so
Well, we've
again.
got Carl sitting as the pilot. We've got Levi
as navigator.
We have Don as co-pilot. We have
want to go in on this?
Tara on video. And we have Dan as video clipper.
And we have a sponge on the rock.
And we have a sponge on a rock.
And we have an anemone on the rock.
And we have a coral on the rock. We have
All
lots of
goodies.
stuff.
Any interest in any of these?
I think maybe just snap zooms on all of these,
please. Go in
please.
video, and I'll just work to the right.
Coral is...
Sounds good.
Oh, I thought Tina went to bed.
Hold
Coral is Bathy Pathies.
Okay.
Oh, this sponge stalk is loaded with, looks like hydroids, but I could be mistaken.
Oh, tiny.
Very tiny little
anemone
coral.
is here.
that, an enemy has a long stalk.
It does.
Ish. Oh, there's another coral right below it. To the left.
Okay.
The other left.
None of that was a beauty shot. Oh.
Below,
that
and
one.
to the left
Yeah.
anemone is the one I meant.
Yep. Got it.
You know.
Okay.
Okay, what's on the
And
right?
we're seeing some more of those barrel-like,
oh yeah, the
cylinders.
sponges.
The chopped off cylinders.
Looks like ship just got in position. Got about a couple minutes to swing and
Roger.
see what we got.
We're at 1,940 meters on Friar Guyot. Gio.
You'll learn.
Gio.
No
One day
T.
I said Gio without the T, and somebody corrected me and said Guyot. So,
Nope.
Gio.
If you ever set foot on the Princeton campus, you would know Gio Hall is the one that houses the geology department, and an Allosaurus, which gets decorated whenever there's a football game.
I set foot on the Princeton campus, but it was many years ago when I was in college, so I probably wasn't looking for...
You weren't looking for rocks?
I wasn't.
What's the matter with you?
I went to an all-girls school, what can I say?
Oh, okay.
So our path takes us along this wall a bit.
Looks that way, yeah. That's
Yeah.
the heading.
I'm losing my stars here, Shirley. Okay.
So I have a pretty steady, I don't know, 20 to 40% to starboard. Maybe just 20. Now that I'm centered up.
Wow. Whoa, really?
So the way these missions
we should start
operate
another move, I think, too.
is
Yeah,
quite a
ready
bit
for
different
move?
from
Yep.
the way
Let's do
we
it.
typically do research expeditions. This is an exploration
Seems
mission.
to be upward a little too.
And we
Bridge, RV nav.
plan these
Go ahead,
first of all,
RV.
based on NOAA
Yeah,
priorities.
quick another move,
In
same
this
as last.
particular case,
30
this
meters,
campaign is based
bearing
on
310
NOAA
degrees,
priority and
0.2 knots.
responsibility for managing our
30 meters
national
bearing
monuments.
310.2. Is that correct?
That is correct.
And so what
Stand by.
happens is the science community and the resource management community get together, and we decide on some sites that we would like to explore, and we have to justify why we are interested in exploring these sites. They need to meet the mission priorities, and so the number of priorities that we can tick off helps us to kind of prioritize the sites that have been
RV nav
proposed.
bridge move
And
has been initiated.
we get together on a number of
310.2 knots.
meetings, conference calls, and so on
Thank you, Bridge. Move initiated. Good copy.
to finalize our mission plan, expedition plan, where we're going to be diving. And then through telepresence, so that's the technology that makes what you at home are able to see and hear right now. I mean, just in near real time with just a few seconds delay. We're not able only to reach about, well, anybody around the world who wants to log into the Ocean Explorer website, but also dozens of scientists with varying, with expertise in varying disciplines. Yep. Patty wants to say something. Go ahead,
Yeah,
Patty.
I do. What we're looking at on the right side of the screen looks for all the world like pillow lava tubes in this crack in the side of the seamount. So I am wondering, watch lead pilot.
Yes. What do you want to do?
Go ahead, watch lead.
Okay, pilot, I wonder if you could just move a little bit to the right and take a look at these things that look like pillow lavas. They're all covered with manganese, but the form is very suggestive.
Certainly can. We're going to have a new pilot take a look at them for you.
Okay. Oh. Uh-oh. We're getting Master Co-Pilot Don shifting with Master Pilot Carl. Okay. Don, let's look at pillow lavas. And Don is late of Harbor Branch as well, right?
Yes, Don used to be the chief submersible pilot of the Johnson Sea Link submersibles at Harbor Branch.
And
Oceanographic.
Excellent pilot he
I
is.
am on.
He's a
And
new
coming off
deep
Joy
discoverer
Lock now.
That's right. Yep.
But
First
an
time he
old
sailed.
JSL
So let's see
pilot.
what happens.
First time he sailed on the Okeanos
Auto
Explorer.
heading also
And he's
off.
my husband.
It's
Which
still
makes
on
it
there.
How
nice.
was that? Yeah. This worked out
Takes a
really
while,
well.
There we
Wasn't
go.
planned this
Okay.
way. I mean, it was planned that we
Yeah.
got married, but it wasn't planned that he would be on
hope
this
so.
trip.
Oh,
Lordy, lordy, lordy.
So the current will probably just spin you. If you just go off stick, it'll just spin you right
Okay.
into
Ooh,
it.
look at that. Look at that rock. That,
But we wanted to be on a 310 roughly.
Yeah.
okay. So we want to go over more towards
More to the starboard
right.
and look in that crack.
And
Sort of
the joystick
sort of
is
a little
up
valley.
is up.
On
Up
the
is
side
up.
of
Yeah.
these
Up is up. There you
Yeah.
go.
right rocks.
Just took me a second to realize that.
Yeah, what I was doing there, yeah.
Yeah.
They are sort of tubular, aren't
There you
they?
go.
They were.
And
But, you know, we've seen a lot of things that do look kind of tubular. If you just
normally
get a
we
bunch
have
of
the
rocks
Z-
in a
if there's
line,
no current,
eventually
the
the
Z-bias holds
manganese
you perfectly
crust
still. You
will
don't need
come
much vertical.
join them all and
but with
just
it kind
sort
of rolling along
overwhelm
today,
the
you bounce
entire
up and down a
surface.
bit.
Okay.
So I just kind of, if I see in my cameras
you
I'm
wanted
getting
to go
too
over to the
low, or I
right.
see my altitude get too low, I just give little pulses up so I don't. Yeah. So yeah, Patty wanted to see rocks off to the right side
Off
there.
to the right.
Yeah.
Okay.
So those little projections that are coming up from the rock look like they may be the bases of those stalks that we're seeing toppled over and
really
buried in the
the
sediment.
Scattered all over the
You can see your
yeah.
toes
slope
and you can see the
below
rocks
us.
in the shadows.
Just
Oh,
lots
yeah,
and
yeah,
lots
you
of
can.
it.
That's good. Thank you for pointing that out.
Yeah, definitely for the
Yeah.
awareness. I like flying by those.
Yeah, it gives you a little better sense of where the bottom is than this other camera.
Exactly, yeah.
So this big pile up here, is that what we're looking for?
Kind of in the depression. Right, Patty?
Yep.
Further right.
In
if
the
you just
depression.
kinda
Well,
lateral.
there may not be a depression
Yeah, there
here.
may not be
Although
a
I'm moving.
Well, it
depression.
looks like there might be, or at least something behind that ridge that's to
Ship's
the
almost
right
in
of
position.
where the
Probably got a
sub is
couple meters
headed.
left.
Okay.
I don't know. The thing I was wondering, thinking about it, and we
So you're
know
probably
that
faced
there
right
is
into the
faulting
current
on
now, which will be a
this
nice way to
half
drive.
Yeah.
of the crest of the seamount. And I just wondered if during that process there may have been some subsequent volcanism, because there are a series of volcanic cones at the top of and on top of the flat part of the seamount. And as you look from east to west, they are lined up almost in a
Sirius
straight
view.
line
Yeah.
if you look across the top of the
Interesting.
seamount.
Do you see
And
the Sirius
those
view
must
there?
have
Yes,
formed
I did.
after
Yeah, looks like a-
the erosional
looks like
process
walls
that
that have
just
been built there,
flattened
huh?
off
Yeah.
the top of the seamount.
It's pretty cool.
Weird.
So in the Sirius
wanna give
view,
a
you
little
can see
zoom?
that what we're approaching is
Sirius?
basically a little trough. We're looking at the far end of it at the top of the field of
good.
view, so
Thank you.
that's a little
yeah.
bit of a scarp.
Oh, yeah, if you look at it in the series view, you can see that. Yep.
And
sure.
so
I
that
zoomed a
would
little
be
on
the
Sirius.
north
I'm not really that
wall
close to
of a little trough, and the bottom of the trough looks very flat. So if the pilot were to turn to port now, we could
Turn
sort
to
of
port.
fly
Okay,
up
roger.
this
So
little
ship is
trough
stopped.
and see if we see
Co-pilot?
any more
We're
pillow-like
experiencing maybe a
structures,
5-minute
which I
delay.
don't see. It just
Okay,
looks
so
plain flat
give it a few
in
more
this little
minutes.
trough.
Yep.
Oh, that's pretty cool.
Interesting, though, huh?
So, watch lead, you just wanted to sort of follow this little trough here?
Yes, please.
Okay.
Pilot, just follow the trough. We're looking to see if we see any
The trough
pillow
happens
tubes
to be going
in here,
310
but
also. So.
it's kind of petering out now.
Yeah,
There
is.
were some a little bit lower down.
Kind of hard to tell with all that manganese.
Yeah, that's true. Well, like I said, you can see here on the edge
you
of
can go wide on Sirius now.
the wall at the top right of the screen, all of those rocks have been covered so fully with a thick coating of manganese that they look as if they are one single long, smooth, round outcrop.
Yeah, I can
But
feel
that was
that
interesting.
pushing me off to the port too, yeah.
Yeah, if it does get really stronger than this, sometimes we just face it in lateral.
Yeah.
Which is, you can also do that anytime if you want.
What's that up on the rock? Is that something there? Anybody wants
There's
to look
something.
at that?
Yeah, we can take a look at that.
Of course, if you're facing it lateral, you have to make sure you don't lateral into anything.
Yeah.
Do you have the swing arms up?
Oh, it has very funny morphology.
Is this of interest or not?
What do you think, Shirley?
Well, we can take a quick
quick snap.
Quick snap on it.
Looks like there may be a brittle star on top.
And co-pilot, when you come in on something like this, you typically set down and put a little thrust down as well?
Yeah.
Or,
It looks like you're flat, so you just, thrust right down.
Just thrust down right there, and then pivot on that.
Yeah.
And that you have to hold in, right?
Depends. You're fairly faced into the current, it might. But yeah, you keep a little active heading on usually.
I'll zoom in.
So Chris is calling this
I'll tip up. Oh, okay.
Roger.
And if your footing's
Ooh,
unstable
very
when you go to
good
zoom,
one.
you lose your
Yeah,
heading.
dead.
Which is when I then ask
Now, is
co-pilot
that a
to
C
tilt.
star or a brittle star? Look at the projections on the arms.
I'm going for brittle star, but I don't know.
Yeah,
Diva
clear.
says a sea star.
Okay.
Yeah.
Moving on.
Okay.
Yeah, moving on.
Thank you.
You're welcome. It's good practice for me.
Yeah.
Ship stopped
Give
4
me the
minutes
easy
ago.
stuff first.
Okay.
You ready for our next move, co-pilot?
Yeah, you ready to get ahead?
Yeah, you want me to push out here?
Yeah, if you push out
up
in
to
center.
310? Right.
Yeah. The current's slowly been working you over to the port side of my screen.
All right, I'll call it in.
All right.
Ready to draw RV nav.
Go ahead, RV.
Ready for another ship move, same as last. 30 meters, bearing 310 degrees, 0.2 knots.
Let me read that back. I've got 3
So if you just, you're still kind of sliding to my port.
Good
if
guy.
you just
lateral
turn, or you could just turn into the current and drive across my screen.
All right.
That's actually easier in a way. And you don't want to get too much farther forward, or you'll get to the end of your tether, and then it gets even harder to hold heading. You know?
What do you think looking at this, Patty?
Well, it's a little outcrop.
Very little.
It is
low. It's a low.
Low.
It's an outcrop head.
Yep, that's true. I think I wrote in the chat log it they're outcroplets.
You're probably feeling a little tether, I guess.
Okay.
Yeah.
Because when you turned, it actually pulled your tail a little farther from me, so I guess you're right on the edge. So you could also sidle to starboard.
Okay.
It's been
take
a long
that.
dive.
If you watch my heading, it's about 20 degrees off now because I'm fighting. So
Wow.
it's always easier for everyone if you.
Boy, the
stay
formation
in the middle.
of Manganese oxide is incredibly complicated, folks.
Patty's been doing some literature search.
Yeah, I have. And
so now come to my heading, or just come over some more?
Many different organisms have the ability to catalyze manganese oxidation.
Light pool area.
A diverse array of everything from bacteria, fungi, algae, and even eukaryotes.
But you've got plenty of tether now.
Okay.
Among the microbial things, the prokaryotes have the ability to oxidize manganese, and that is quite widespread. We got
to the left.
Cyanobacteria.
The big stalk thing or the whip? Both? Whip first. Okay.
Diversity of heterotrophic rods and cocci. The shielded and budding bacteria. Some purported autotrophic strains related to Pseudomonas species. And the Metallogenium group. Aerobic lactobacilli use the manganese oxidation reaction as protection against oxygen fugacity.
Okay, so we have, is this a bamboo? This is a bamboo. Very long whip.
Okay.
Yeah, oddly enough, Les wants to look at this.
I can't imagine why. Can you, Chris?
Actually, you could just tilt up.
Stay here.
Oh, no, can't even think of that. Can't even imagine.
Oops, too far.
Okay, we might have to twist. You're right.
Okay. You want the lasers off?
It's like me wanting to look at glass sponges. It just doesn't make sense, does it?
Not at all.
Yeah, so give it a little twist to port.
Yep.
And if you want, I can take the camera while you hold it steady.
No, let me try it if it's not a big deal.
Yeah, it seemed pretty stable.
Okay, zooming a bit more.
Yeah, a little bit more.
Okay.
Nice.
Beautiful polyps. So on each of the tentacles, there are little tiny projections. Are they like pinnules, or what is that structure called?
Okay.
Okay.
I'll get a quick establishing. Just like that.
Okay.
And we're clear.
Okay, watch lead.
Yes, pinnules, good. They are pinnules. Okay, yeah. Yeah, Don, if we could go look at the one in the back.
In the back? The whip
please. Yeah,
up?
The whip standing straight up. Yep.
So you'll have to choose your approach so you don't run over this one now.
Yeah.
Oh, yeah? Okay.
Yes.
Let's see, what's over that one? Is a big rock. It's not too big. It's all right.
Yeah.
So Scott France remarked that this is that skeleton on that long whip that we just looked at, and probably the one that we're going up to as well, is thin. It's thin for the size of the colony. We're going to take a look at this one coming up.
And this would be good too, because you're into the current, and any sediment we stir up is going down.
Yeah.
Downwind.
So it's good.
So, yep, always good. Sep will be up on a rock.
Which could make you a little bouncy, but
yeah,
it'll work. You might have to actively hold your heading in with little lefts and rights.
Okay.